Hello students. In the today's session we are going to discuss a very important topic of pharmacology, a fundamental concept of pharmacology, that is the cardiac electrophysiology. Now cardiac electrophysiology is a science that deals with the study of electrical activities generated within the heart. Now we all know that there are two types of cardiac cells. One are the specialized cardiac cells called as the pacemaker cells, and the other are the normal typical cardiomyocytes, that is the contractile cells. So today we are going to, in this session we are going to discuss the cardiac electrophysiology of typical cardiomyocytes, that is the normal cardiac cells, that is the normal cardiac contractile cardiomyocytes. Now under this topic we are going to study 4 subtopics. First and the most important of all is the generation of action potential, how there is generation of action potential in the normal typical cardiac cells, in the normal typical contractile cardiomyocytes. Then second subtopic is the conduction of action potential. Third is the excitability, how these normal cardiomyocytes they get excited. And the fourth is the generation of a refractory period. Now let's discuss in brief about the properties of cardiac muscles and the functioning of heart, that is the conducting system of heart. Now there are 4 cardinal features of cardiac muscle tissue, and these 4 features or the properties are the most important of all, that is the automaticity, then conductivity, rhythmicity, and all or none phenomenon. Now this is the diagram. This diagram shows the wall of heart. This is the heart and this is the wall of heart. The wall of heart is made up of cardiac cells termed as to be the myocardium, and myocardium is made up of 2 types of cells, normal cardiac cells and specialized cells called as the pacemaker cells. Now these pacemaker cells are like the SA nodal cells, the cells of sinoatrial node, they are specialized cells called as the pacemaker cells, and the cells of SA node they are termed as to be the pacemaker cells or the nodal cells, and they possess the property of automaticity. So this is a property of automaticity which is possessed only by the pacemaker cells or the nodal cells like the cells of sinoatrial node. Now automaticity is the intrinsic or the natural ability of heart to depolarize itself spontaneously without any external stimulus and trigger an action potential. Now this action potential, it is triggered in all the directions of the heart. The cells that is the pacemaker cells, they get depolarized and they trigger an action potential or they generate an impulse, and that impulse or that action potential is triggered in all the directions, and since it is triggered in all the directions, it is the heart that contracts, the whole heart contracts as a unit. Now this is the diagram that shows the conducting system of heart. Now the elements of conducting system of heart are the sinoatrial node, then atrioventricular node, bundle of His, then the bundle branches, that is the left bundle branch and the right bundle branch, and the small fibers called as the Purkinje fibers. Now we'll talk about these elements one by one. Now first of all is the SA node or the sinoatrial node. Now SA node depolarizes and it generates the action potential or impulses. Now impulses, they are generated at a rate of 60 to 80 impulses per minute. Now this SA node, it is located in the wall of the right atrium, and it is called as the pacemaker of heart because it is the SA node that generates impulses or the electrical signals. Now impulses from the SA node, they sweep over the entire wall of the atrium so that the 2 atria they contract at the same time simultaneously, and the time taken by the action potential generated by the SA node to reach the AV node is about 0.1 second, and that is the time taken by both the atria to contract. Now the impulse from the SA node reaches to the AV node. From the AV node, the impulse travels to the bundle of His, and from the bundle of His, the same impulse which is generated by the SA node, it travels to the two bundles, that is the left bundle as well as the right bundle, and finally that impulse is transmitted to the Purkinje fiber. And this results in the transmission of impulse from the AV node to the apex of ventricular myocardium, and this results in the contraction of both the ventricles. So contraction of atria is followed by the contraction of ventricles, and the entire heart contracts as a unit. So this property of transmission of impulse which is generated by the SA node throughout the atria and the ventricles is called as the conductivity. That means a single impulse which is produced by the SA node, that single impulse is responsible for the contraction of atria, both the atrias, and the contraction of both the ventricles. One impulse and one contraction of the heart. Now, third property is the rhythmicity. Property of automaticity and conductivity leads to the property of rhythmicity. That is, the heart contracts in a rhythm. When one impulse is generated by the SA node, the entire heart contracts once. And on an average, SA node generates 72 impulses per minute, and as the heart contracts or beats 72 times per minute. So this property of the heart, that is the heart always contracts in a rhythm, is termed as a rhythmicity. Now the fourth very important property is the all or none phenomenon. Now a single impulse generated by the SA node, it travels to the single impulse generated by the SA node, it travels to the atrium, that is to the atrioventricular node, and the same impulse travels to the apex of heart, resulting in the coordinated pump activity of the entire heart. That is, the entire heart contracts as a unit. Now either the impulse generated will contract the entire heart and produce a complete response, or there will be no response. That means there will be no pumping of the heart. So this property, that either the heart contracts as a unit or the heart does not contract at all, is termed as all or none phenomenon. In order to further understand cardiac electrophysiology, let's see the characteristic properties of pacemaker cells and normal myocytes. Now, as we have already seen, myocardial tissue is made up of cardiac cells. Cardiac cells are of two types. Specialized cardiac cells or myocytes are the pacemaker cells, and the normal myocytes are the normal contractile cells. Now, the main function of the pacemaker cells is to depolarize, and when these pacemaker cells depolarize, they generate an impulse, or in other words, we can say that they trigger action potential. So the main function of pacemaker cells is to depolarize and generate an action potential. Pacemaker cells are responsible for the contraction of the heart as a unit. Now, when we talk about contractile work cells, they depolarize and they contract. So the main function of contractile work cells is to depolarize and contract. They are responsible for the contraction of heart. Pacemaker cells, they possess the property of automaticity. That is why they intrinsically, on their own, naturally they depolarize. Whereas the cardiac myocytes, normal cardiac myocytes, they depolarize only when they are stimulated by the pacemaker cells. Now, when we talk about the action potential of pacemaker cells, action potential is slow and short action. That is, the duration of action is action potential is short. Now, this is the diagram. Here I have shown the action potential of pacemaker cell. As we can see over here, depolarization occurs slowly and duration of action potential is very short. Now this is a figure that shows action potential of normal cardiac cells, normal contractile work cells. Here we can see that there is rapid upswing of action potential, that is depolarization is very fast, and further there is a plateau phase. Because of the presence of this plateau phase, duration of action potential is long. So in the pacemaker cells, depolarization occurs slowly and duration of action potential is short, whereas in the normal contractile cells, there is rapid upswing of action potential, depolarization is fast, and duration of action potential is long. Now, a very important characteristic feature of cardiac cells is the presence of gap junctions. Now, this is the diagram. In this diagram, I have shown a series of cardiac cells. Now, the first cardiac cell that is shown over here is the pacemaker cell, and the other cells are the normal contractile cells. Now, this pacemaker cell, when it depolarizes, it generates electrical impulse, or it generates action potential, and this generation of impulse or the action potential occurs due to the movement of ions. Now, this action potential which is generated by the pacemaker cell, it is further propagated to the adjacent myocytes by the diffusion of ions from the gap junctions. So, when there is diffusion of ions from this pacemaker cell in the adjacent cardiac cell, this cardiac cell gets depolarized. Further, when this cardiac cell depolarizes, there is movement of ions from this depolarized cardiac cell to the adjacent or the adjoining cardiac cell. So this way, because of the movement of ions, specifically the calcium and potassium ions, due to the diffusion of ions through the gap junctions, there is propagation of action potential. So action potential wave propagates by diffusion of ions through the gap junctions. Now we will talk about action potential in a typical cardiac cell or a normal cardiac cell, that is the contractile cell. Now, as we have already seen the role of gap junctions, these cardiac cells are stimulated because of the diffusion of sodium and potassium ions inside these cells through the gap junctions. So, these contractile cells, they are stimulated by the diffusion of ions, that is the diffusion of sodium and potassium ions inside these cells, and these ions, they diffuse or they come either from the pacemaker cells which have been depolarized, or from the adjacent normal cardiac cells which has undergone depolarization. So diffusion of sodium and potassium ions in these contractile cells results in their stimulation. And the action potential of these normal cardiac cells show 5 phases. Now, as shown over here in this diagram, there are 5 phases characterized as phase 0, 1, 2, 3 and 4. Action potential begins with the phase 4 and it also ends with the phase 4. Now, this is a phase which is shown in the blue color. This phase is a phase 4. This phase is called as a resting phase, and during the resting phase, the cardiac cell, the normal cardiac cell, it depicts a potential of minus 90 millivolts. And when the cell is in the resting phase, the cell shows normal polarity. Normal polarity means that there is higher concentration of potassium inside the cell, and there is higher concentration of sodium and calcium outside the cell. So the cell, when is in the resting phase, the cell shows a membrane potential of minus 90 millivolt and normal polarity. Now, as soon as there is diffusion of sodium and calcium ions inside the cell through gap junctions, there is rise in the membrane potential from minus 90 millivolts to minus 70 millivolts. Now, minus 70 millivolts is the threshold membrane potential. It is the membrane potential at which action potential is triggered. And as soon as the cell acquires this membrane potential of minus 70 millivolts, there is opening of voltage gated fast sodium channels. And because of the opening of these channels, there is rapid inward current of sodium ions. And because of this rapid inward movement of sodium ions, the membrane potential further rises from minus 70 millivolts to minus 40 millivolts. Now, as soon as the membrane potential rises to minus 40 millivolts, there is opening of L-type calcium channels and there is influx of calcium ions inside the cell. And this results in further rise in the membrane potential to plus 10 millivolts. So, very important to note over here is this, that the membrane potential rises from minus 90 millivolts to plus 10 millivolts. This phase is called as the depolarization phase. Now, in the normal cardiac contractile cells, this depolarization phase is very rapid and there is predominant role of sodium ions compared to the calcium ions during the depolarization phase. Now, next phase is the phase 1. Phase 1 is the phase of early repolarization. Now, in this diagram, this phase 1 is shown here in the black color. During the phase 1, there is an activation of sodium channels. And during this phase, there is fall in membrane potential from plus 10 millivolts to 0 millivolts. Now, what happens during this phase is this, that there is inward movement of calcium ions and there is outward movement of potassium ions. But because of the higher outward movements of potassium ions and less comparatively less influx of calcium ions, there is fall in the membrane potential from plus 10 millivolts to 0 millivolts, and this phase is called as a phase 1. Phase 1 is followed by a very important phase that is termed as a plateau phase, that is the phase 2. It is shown here in the red color, and this phase 2 or the plateau phase is responsible for the long duration of action, action potential of normal cardiac cells. Now, let's see what happens during this phase. During this phase, there is inward movement of calcium ions, there is outward movement of potassium ions, but potassium and calcium currents in opposite direction balance each other. That is, influx of calcium ions is more or less balanced by the outward movement of potassium ions, and that results in the plateau phase. There is not significant change in the membrane potential, and thus we can say that the membrane potential remains somewhat stable for about 200 milliseconds, and this prolongs the action potential. And this influx of calcium ions is also responsible for the further release of calcium ions within the cell from sarcoplasmic reticulum, and this calcium induced calcium release from the sarcoplasmic reticulum inside the cell is responsible for the contraction of cardiac myocyte. So, the contraction of cardiac myocytes occurs in the phase 2, that is in the plateau phase, and this occurs because of the release of calcium from the sarcoplasmic reticulum, that is the calcium induced release of calcium from sarcoplasmic reticulum. And because of this plateau phase, which remains for about 200 milliseconds, there is prolongation of the duration of action potential of a normal cardiac cell. Now, phase 2 is followed by the phase 3. Now, in this figure, the phase 3 is shown here by the black color. The phase 3 is the repolarization phase. During the phase 3, there is an activation of calcium channels, so there is no movement of calcium ions, and there is persistent outward movement of potassium ions. That means there is persistent leakage of potassium ions outside the cell, and because of the loss of these potassium ions, the membrane potential falls to minus 90 millivolts. Now, at the end of the phase 3, the normal membrane potential of the cell is attained, but the polarity of the cell is reversed. That means earlier in the phase 4, there were more of potassium inside the cell, but at the end of phase 3, there is more of sodium inside the cell and there is more of potassium outside the cell. Now, during the phase 4, the resting membrane potential is minus 90 millivolts and the normal polarity of the cell is also achieved by the activation of sodium potassium ATPase pump. Now, what this pump does is this, that it throws away excess sodium ions outside the cell and it pulls the potassium inside the cell. and for every loss of 3 sodium ions, there is gain of 2 potassium ions. So at the end of the phase 4, the membrane potential becomes normal, as that has already been achieved at the end of phase 3. So at the phase 4, the membrane potential is minus 90 millivolts, and the polarity of the cell also becomes normal. Because of the activation, because of the functioning of this sodium potassium ATPase pump, the cell achieves the normal polarity, that is higher concentration of potassium inside the cell and higher concentration of sodium outside the cell. So this is how a cell from the resting phase, it gets depolarized. Followed by that, it enters into the plateau phase where it gets where it contracts, and again it comes back to its resting membrane potential, and it also achieves its normal polarity. So this is the action potential. Now action potential is followed by the conduction. Now action potential, which is generated in a normal cardiac cell, it is propagated to the adjacent cardiac cell. Rate of conduction depends upon the rate of rise of action potential. Higher is the rate of rise of action potential, higher will be the rate of conduction. Now in the normal cardiac cells, this rate of conduction, or the rate of conduction is high because of the higher rate of depolarization. So there is faster conduction in the cardiomyocytes of atria, ventricles and Purkinje fiber, and conduction velocity is found to be 0.5 to 5 meters per second. Excitability of normal cardiac cells. Now as we all know, diffusion of sodium and potassium ions inside the normal contractile cells result in their stimulation or excitation. So typical contractile cardiomyocytes are stimulated by diffusion of sodium and calcium ions through the gap junctions due to depolarization of adjacent pacemaker cells or the adjacent cardiomyocytes. Next is the refractory period. Now refractory period is the period of time during which the threshold for producing an action potential is increased. Now refer to this diagram. There are two types of refractory period. One is the absolute refractory period, which is shown here in the purple color. This is the absolute refractory period. Now during the absolute refractory period, action potential cannot be produced irrespective of the strength of stimulus. So during this period, action potential cannot be produced. Another refractory period is termed as to be the relative refractory period. Now here during this refractory period, action potential can be produced, but the stimulus should be very strong. The strength of the stimulus should be much more higher compared to the normal stimulus. So stimulus stronger than the normal stimulus can induce the action potential. So this is all in detail about the cardiac electrophysiology of normal typical contractile cardiac cells. I hope you must have understood the cardiac electrophysiology. Thanks for watching the video.